In this video, we're going to look at the structure of skeletal muscle fibers, which are skeletal muscle cells. And they're called fibers because they are such elongate cells. They're very long. As we can see in panel A here, a photomicrograph, these cells, I'll outline one here, are formed by cell fusion during development. And so they may have, not shown here, but they may have hundreds of nuclei and literally thousands of mitochondria. So these are huge cells. They can be multiple centimeters long, almost as long as the muscles that contain them in many cases. Now let's look closely at a couple of things that we see here. One of the key characteristics of skeletal muscle is that it is one of the striated types of muscle, right? Cardiac being the other. They're labeled down here. I'm going to circle some up here, right? Circle this little area right here. And if you look very closely, there's little tiny stripes or striations. That's literally what the word striation means, is stripe. And those are generated by repeating units of protein filaments that are going to generate our muscular tension. And we'll look at those in just a moment. Something else we see here are the neuromuscular junctions. We see several of them, actually. Right? There's one here, there's another one over here. These are enormous synapses that are meant to convey a stimulatory signal. In other words, a single action potential traveling along the axon here, right, down to this motor end plate, the synapse. Because of this enormous surface area, we get a very large stimulatory depolarization of the skeletal muscle fiber. And then because the skeletal muscle fiber possesses voltage-gated channels, we will get an action potential that will run outward from this location along these skeletal muscle fibers as they are activated. Now, if we come over here to B, we see a cartoon version of some of the same structures. We see our motor neuron here, right, innervating our skeletal muscle fiber. Here's our neuromuscular junction. And so we would get a depolarization due to acetylcholine release, right, in this little synaptic cleft here, very large synaptic cleft actually. And then, as I mentioned, we would get an action potential that would travel outward from this because of voltage-gated channels. Now, what we see is that the membrane of the muscle cell, referred to as the sarcolemma, invaginates, right? We have these indentations, deep indentations into the muscle fiber. And that allows our action potential to actually travel down deep inside this cell. Now, the inside of the T-tubule is still outside the cell. It's just an indentation. And so it contains extracellular fluid. So we can get an action potential that literally travels along that membrane of the T-tubules deep into the cell. And that's really important because it delivers the stimulus to this giant cell deep inside, where it's required to elicit activation of all of these protein filaments that are going to generate our tension. Right? And so what we see is that our T-tubules are actually surrounded by what's called the sarcoplasmic reticulum, this membrane-bound organelle, which is literally just the endoplasmic reticulum of a muscle cell. And it is a storage depot for calcium. And so what we're going to actually find more detail on later is that our action potential is going to stimulate calcium release from these storage sites, right, inside this sarcoplasmic reticulum. And the calcium is actually the trigger for tension development in our sarcomeres, right? The sarcomeres being those repeating units of proteins that are sort of repeat over and over and over again. So here's a sarcomere, right, one end to another, right? But we've got another one that starts right here and then continues this way, right? And another on this side that goes this way. And so our myofibrils, right, there's our myofibril over here, are simply bundles of sarcomeres lined up end to end to end. And sarcomeres are a couple of microns long, and so, right, millionths of a meter long. And so we have thousands of these end to end within each myofibril. And then, of course, the muscle cell itself is basically a bundle of myofibrils with all the accessory structures that a cell requires. Now, if we come over here to C, we'll take a quick look at the sarcomere, and we can see that we end up with these darker regions, right, the A band, and then lighter regions, the I band, and they repeat. right? Light, dark, light, dark. Well, that gives us this striated or striped appearance that we saw up here, right? These light, dark bands, the striations, are caused by regions of overlap and non-overlap between those protein filaments that are going to generate our contraction tension.